The next speaker is Yaojiong Wu from Tsinghua University, China. His talk is Inflammatory Factors in Hair Follicle Regeneration.
So good afternoon, Chairman. Good afternoon, everybody. My name is Yaojiong Wu from Tsinghua University in China. First, I would like to thanks to the meeting committee for giving me the opportunity to share our experience in our recent study on inflammation in hair regeneration, particularly in the hair follicle de novo regeneration after wounding. So now we all know there are stem cells in the skin. For example, in the visual epidermis and in the body region of the hair follicle. So both populations of the stem cells are able to form de novo hair follicles. So one population of the cells in the stems in the hair follicle can be labeled by LGI5. So however, after the wounding of the skin, the hair follicle and other appendage structures are not regenerated. So this suggests that some environmental cues are basically inhibitory to the hair follicle stem cells or the stem cells in the other stem cells in the skin. So we tried to test if these stem cells are really able to regenerate the hair follicle. So we isolated the epidermal stem cells, which are labeled in red, and the skin cells in the dermis. And after culture expansion, we mixed them in the hydrogel. So after culture, we can see these cells have intrinsic ability to migrate to their anatomic position. So the epidermis stem cells migrate up, and these dermal stem cells migrate down. So after transplantation to the excisional wound, so we can see the cells form nice hair. Histological analysis indicated that this eugenic epidermis and the eugenic hair follicle are formed by the transplanted stem cells. So the dermal stem cells formed the DP cells in the hair follicle, as well as numerous dermal cells in the dermis. So the regenerative structure also included this species gland. So why these structures are not regenerated after wounding? So we try to understand what happened in the wound. So the first thing that we thought out was inflammation. This is a significant event that occur immediately after wounding. So numerous inflammatory cells infiltrated the wound. So previous study suggests that inflammatory cells are likely to play an inhibitory function to the regeneration. For example, studies indicate that the inflammatory signals are going up with skin aging. And also some other studies indicate that depletion of some inflammatory cell populations actually do not affect the speed of wound healing, even accelerate wound reactivation after depletion of neutrophils. And in some other organs outside of the skin, for example, in the heart, depletion of some inhibition of some inflammatory cells to the ischemic myocardium actually can reduce the influx size in mice. So these studies suggest that the inflammatory signal may largely play a suppressive role in skin regeneration. And another evidence says that the wound healing in the human fetus, particularly in the early stage, so after deep wounding to the fetal skin, so the skin is can be fully regenerated without scar formation. And in the wound healing process, so the very lower level of inflammatory cells are found and infiltrate the wounded tissue, so which is very different from that happened to the adult wound in the skin. So however, there are different results from recent studies. For example, from Kasuri's lab in the University of Pennsylvania, which discovered that actually in mice, the hair follicle can be regenerated after wounding. So, which is mediated by subpopulation of T cells which release FGF9, and FGF9 being stimulated, induce the upregulated dispersion of Wnt in the dermofibroblasts, and finally ended up with this de novo follicle regeneration. So this study and some others suggest that some factors in the wound might be necessary for the regeneration of the skin structure, including the hair follicle. So this makes it quite complex to understand the impact of inflammatory cells in the hair follicle regeneration. So the hair follicle, particularly the hair follicle undergo cyclic regeneration, and in mice, these three stages can be clearly defined, and which have a long telogen stage, start from around 7 weeks, and this provides us an ideal model to study the stem cell activation in the hair follicle. So now we know that the hair follicle stem cells are suppressed by the BMP signal in the telogen stage. So when the hair follicle goes to the energy stage, so the suppressive signal from BMP must be suspended. So however, in the wounding model, so it looks like this suppressive signal can be overridden. For example, in the wounding, for example, the excision of wounding to the mouse skin in the telogen stage, which can induce the hair follicle to regrow in the tissue surrounding the wound. So this can be indicated in a histological analysis where we can see that the hair follicle stem cells are activated, they are proliferate, and the hair follicle are elongated. So now this model suggests that after wounding, so there are some molecules which are upregulated and diffuse into the surrounding tissue, so which activate the stem cells in the hair follicle. So we use this model to try to define which factors might be playing such a role. So firstly, we examine if it is if it is the effect of inflammatory cells, so we use the dexamethasone and FKB inhibitor to inhibit inflammatory cell infiltration after wounding to see if this actually activates wounding-induced hair follicle antigen. Actually, they did. Both application of either dexamethasone or FKB inhibitor completely attenuated wounding-induced hair follicle antigen. So the next, we tried to understand which cell population of inflammatory cells played the role. So we injected neutrophils and activated and activated macrophages into the skin, and found that the injection of activated macrophages into the skin induced the hair follicle into the anagen stage, and the hair regrows. So the next we try to understand which subpopulation of macrophages actually play a role. So the next we depleted macrophages in the mice. We use chemical to deplete macrophages in mice, and found that the depletion of macrophage completely abolished this wounding-induced hair follicle antigen. So there are two subpopulations of macrophages in mice, which are called inflammatory macrophages and the tissue resident macrophages. The inflammatory macrophages are derived from a monocyte in the bone marrow, while these tissue resident macrophages are considered to be originated from amerogenesis, which can self-renew to maintain the homeostasis of this cell pool. So these two subpopulations of macrophages can be genetically depleted in inducible mice. So the depletion of we found that depletion of inflammatory macrophages in mice can completely attenuate wounding-induced hair follicle antigen, and also the hair follicle regeneration after wounding. While the depletion of these tissue resident macrophages did not affect hair follicle regrowth and hair follicle regeneration. So next we try to understand what factors released by this inflammatory macrophages mediated the effect. We performed the microanalysis of these three populations of immune cells. So we found that 15 secreted factors are highly expressed in this inflammatory macrophage population, which included interleukin 6 and TNF-alpha. So now we try to verify the effect of these upregulated factors secreted by this inflammatory macrophage. We tested a few of them. For example, interleukin 6, we found that loss of interleukin 6 in mice did not affect this wounding-induced hair regrowth, while knockout of TNF-alpha did abolish this wounding-induced hair follicle antigen. And further, we found that TNF-alpha is effect through the TNF-alpha receptor 1, but not receptor 2, to mediate this hair follicle regrowth. So our results are consistent with a study from Dr. Jones' lab in University of Southern California, so which showed that actually hair plucking injury could induce macrophage to release TNF-alpha, which mediated hair regrowth. And we found that use luciferase TNF-alpha mice, we found that TNF-alpha, the expression of TNF-alpha maintained a high level after wounding in the wound. And the loss of TNF-alpha in mice dramatically reduced hair regeneration in the wound. But to our surprise, where expression of TNF-alpha actually completely inhibited hair regeneration in the wound. So this is something we didn't expect. This result, I think, we will suggest that TNF-alpha actually is in a high level already in the wound, and excess TNF-alpha might not helpful to wound regeneration, to hair follicle regeneration in the wound. So next, we try to understand which intracellular signal downstream of TNF-alpha mediated the effect. So we performed a phosphorylation array analysis of the stem cells treated with TNF-alpha. We found that the TNF-alpha and we found that some factors, we found that some proteins are induced to phosphorylate after TNF-alpha treatment. So bioinformatic analysis only enriched this phosphorylation to signal of AKT, of PI3 kinase AKT. So when the epidermal stem cells are treated with the TNF-alpha, which induced the phosphorylation of AKT, the same happened in vivo. So after TNF treatment, after wounding to the skin, we found that AKT phosphorylation increased in LGR5 stem cells in the hair follicle. And in induced for loss of chitin, the mice when tamoxifen is locally injected, can induce hair follicle antigen. When tamoxifen is administered systemically, it can induce this widespread hair follicle growth. So now we found that actually the hair cells from the hair follicle, for example, LGR5 stem cells can move upward along the hair follicle to the epidermis after wounding, and this stem cell, the progeny of these LGR5 stem cells can contribute to the neogenic hair follicles in the wound. About 40% of the neogenic hair follicles contains cells derived from the liver hair follicle. And the loss of chitin in LGR5 stem cells could enhance de novo hair follicle regeneration in the wound. So we found that it increased the migration of the stem cells to the epithelialized epidermis and increase the number of these cells move downward to the newly formed hair follicles. So next we try to understand the contribution of neutrophils in hair follicle regeneration. We used functional blocking antibody to deplete neutrophils in mice. And repeated administration of this antibody could deplete neutrophil to less than 1% in peripheral blood. However, unfortunately, we didn't see a difference after in terms of hair follicle regeneration after depletion of neutrophils in mice. So finally, I would like to thank my colleagues in the laboratory. So the data I presented is largely performed by the graduate student of Yaojiong Wu, and the postdocs in the laboratory. And also thank to the collaborators who give some of the mouse lines we used in this study. So that this study was supported by the several funds from the central government of China and the local government in China. Thank you.
Now, the session is open for discussion. Any questions from the audience? Question over here. Okay, beautiful talk, great stuff. Like I share your interest in looking at the immunology of wounding and hair regeneration. And just one quick point on you used the IL-6 knockout mouse to look at ligands, like you looked at the TNF knockout mouse. And just one comment is, although you found that the TNF looked important, but the IL-6 didn't, it looked like it didn't change. What we found with that IL-6 knockout mouse is that there's upregulation of other IL-6 family ligands. And so that actually, if you look at like phospho-STAT3, it's actually higher in the IL-6 knockout. So you might want to look at an IL-6 receptor knockout instead. It might give you a different result.
Thank you for a great talk. When we did the talking experiment we did it before, talking for de novo hair and the crown sensing hair regeneration, we did that. We find out the M1 macrophages involved in this process. And how about the M2, maybe the regeneration process, maybe involved in this process too. How do you think about your wounded-induced hair regeneration? Is M1 or M2 or both of them involved in this process?
So we try to understand the M1, and we use a different way to classify macrophages because we feel the M1 and M2 is quite difficult to understand.
Just inflammatory or resistant?
Yeah, for the definition of these two populations. So I couldn't comment on this.
Okay, so where did you think the macrophage came from? Is from bone marrow derived or the resident macrophage proliferation because of the wound?
So the one that we found that that is responsible for the hair follicle regeneration is the one that derived from the bone marrow.
Thank you.
Are there any more questions? Oh, thank you. So we move on to the next one.